Okay, so good morning, everyone. I'm Harris Lewin. I'm the chair of the working group for the Earth BioGenome Project. I'm very honored, actually, to represent the large group of scientists that's involved in planning and executing this project. And before I begin, I want to thank the Wellcome Trust, Jim Smith, for hosting this event, and Suzanne and her fabulous staff for providing this really beautiful venue for us for the conference. I'd like to thank my co-organizers for the meeting, Jean Robinson, who's also co-chair of the Earth BioGenome Project, John Kress, and Richard Durbin from Cambridge for their efforts in putting the program together. I'd like to thank Julia. Where's Julia? Julia Wilson, who is my partner for the last few weeks, last few months in organizing the meeting. Extraordinary work, even from Crete, from the last week, so thank you for your efforts, even on your holiday. Emily Mobley from Sanger, she's running around the back there she is. And Steve Palmer and the Wellcome Trust and Sanger staffs for their support and all the details, especially for tomorrow's launch event. So I'm going to spend a few minutes this morning just to give you an overview of the Earth BioGenome Project and the goals for the meeting over the next day or two. And as I mentioned, I chair the working group. The co-chairs are Jean and John. And the Earth BioGenome Project working group consists of 22 others. This is not meant for you to be able to see, just to know that there's a large group of really outstanding scientists and scientific leaders from around the world that have come together and really to help put this plan in place for the Earth BioGenome Project. And I'm representing them in my talk today, and I'm extremely honored to do so. So this very simple slide really tells us what the problem is. And the problem is that over the past 200 years, the human population has increased in size more than sevenfold, to the point where before the year 1800, for the tens of thousands of years of human evolution, our total human population size was less than 1 billion. And today, human population size is 7.2 billion, going toward 9 billion in the year 2050. Now, this growth in human population size is directly correlated with a linear increase in extinction rate, to the point where we now know that extinction rates are exceeding background levels by more than 1,000-fold. And this has been called the sixth mass extinction events. And it's projected that by the end of the century that we may lose 50% or more of biodiversity on the planet. And actually, nobody knows what the consequences of that will be. We really don't know what the effect on our planet will be. We believe we're starting to see some of those effects. For example, the anthropogenic effects, the increase in extinction rates are due to habitat destruction primarily, and now an increase in that rate due to global climate change. And we're starting to see the effects, for example, off the Great Barrier Reef in Australia, total collapse of ecosystems, and that is leading to some unprecedented changes in our Earth and in our climate. Now, one of the things that we now know, and a recent study just published a couple of weeks ago by Matt Davis, is that extinction rates are actually outpacing the rate of extinction, outracing the pace of speciation. And depending on where and when these extinction rates occur, we are seeing massive losses at very deep branches, for example, in the phylogenetic tree. So we're losing phylogenetic diversity at an unprecedented rate. And when these losses occur at very deep branches, in fact, every time we lose a deep branch, we're losing hundreds of millions of years of evolutionary history on our planet. So this is sort of the problem that we face and to deal with some of that, with those issues, we have proposed a project which is framed as a grand challenge. We've called it a moonshot for biology. I'll talk a little bit more about that tomorrow, which is to sequence the DNA of all known eukaryotic life, 1.5 million known eukaryotic species in a period of 10 years. And the vision for the project then is to create a new foundation, a new infrastructure for biology that will drive the solutions for preserving that biodiversity that we're rapidly losing, and for sustaining human societies through enhanced delivery of ecosystem services. Now, the goals and the vision, the challenges for the project were all laid out in a paper this past April that we published together, the working group in the Proceedings of the National Academy of Sciences. And the response to this paper has really been, it's been very encouraging. There's been a tremendous public response to it. There's been a tremendous response from the scientific community to it. And this has really given us great momentum going into our launch event. Why do we want it? What are the goals of the project? The goals of the project, first, are to maximize the returns of investment in such a project to human society by providing new resources for producing things such as new biofuels, new biomaterials, new sources of genetic variation for feeding the world, new source of genetic variation for crops and for animals, and the development, identification of new sources of drugs for example, slowing the aging process and treating infectious and inherited diseases. This is, something in it for everyone. Second, of course, to preserve, protect, and regenerate biodiversity, to deal with this problem of species loss. We've lost 52% of the vertebrate population on Earth in the last 40 years, 23,000 species out of 80,000 species that were surveyed by the International Union for Conservation of Nature are on the threatened or endangered list. And finally, and important to most of the people in this room, is to revise and reinvigorate our understanding of biology and evolution. And that is to reconstruct the entire eukaryotic tree of life, and in addition to that, to understand, of course, the relationships between all known living things. So what exactly is the Earth BioGenome Project? The Earth BioGenome Project, as it's come to be, is a consortium of independent partner institutions and large-scale genome projects that have a common goal of sequencing and annotating the genomes of all 1.5 million known eukaryotes and sharing the results freely for the benefit of humankind and our planet. And we have made a lot of progress in the last year at putting those partner institutions together. And again, I know it's hard to see this, but there are 19, well, it's impossible to read it. It's 19 different institutions have executed the Memorandum of Understanding, which basically commits them to the goals that I just described for the project, as well as to open access and free sharing of data, and to the extent possible, to comply with the Convention on Biological Diversity and the Nagoya Protocols. 19 institutions and now 16 different affiliated community and regional national projects, many of which are represented in the room today, and you'll hear about in session 1 of the meeting. What are the goals of the organization of EBP? Planning, coordination, and integration across the projects. We need to have standardization, because if we get to the end of this project and everything, all the sequencing and annotation are done in different ways, it will be like the Tower of Babel. We want to do deep comparative genomics across all taxa, and we need to do things in a standardized way. We also have very significant regulatory, legal, and ethical issues in this project, even moving, transporting certain types of organisms across international boundaries. We have the Convention on Biological Diversity, Nagoya protocols that I'd previously mentioned. For access and benefit sharing, these are significant issues, and we need to be dealing with those up front. Communications is very important, how we represent all of the partner organizations in one very uniform way to the external world about the progress made by the project, as well as internal communications between the groups. And part of that, of course, we hope would be an annual meeting in which all the taxonomic groups working can be brought together to share information. Advocacy is very important for a group like Earth BioGenome Project. We need to be out there advocating for our project, working with government, NGOs, and the companies, private industry to, of course, work on securing the funding and long-term funding, the amount of money that we're actually going to need to get over the goal line for this project. So these are our organizational goals, and we intend then how are we going to do it? The project is organized in a way that will allow us to have achievable milestones over the 10-year period. And the first sort of major strategy that we're going to use is what we call the phylogenetic wave. And it really starts at the level of families. We propose to sequence at reference quality over the first 3 years of the project, 9,300 genomes over 3 years. Might sound like a lot today, but I think it is doable, and we'll get into that in more detail. At the Vertebrate Genomes Project workshop in New York a few weeks ago, I showed that with 10 laboratories producing 6 genomes a week, which Eric Jarvis tells me is, tells us all is now possible, and that he is actually doing at the Rockefeller lab. 10 laboratories doing that at 6 genomes a week will get us over the 9,000 reference genomes in 3 years. So that's a very doable I think. The project design says 160,000 then to 200,000 representative species of the 160,000 genera would be phase 2. These are, at least in the proposal, as it exists today, high-quality draft level assemblies. Those are in years 4 through 7, and then finally in the out years of the project, the complete sequencing of all 1.5 million. Now, we know that there are way more than 1.5 million eukaryote species, but when we hit 1.5 million, we're going to declare victory for this project. And in parallel, wow, okay, somebody else's presentation came up on my monitor here. In parallel, does that mean I have a few minutes? Is that a time signal? Okay, maybe. In parallel to this is another strategy which we call the ecosystem sequencing or Google Life strategy. And that strategy is basically sequencing all organisms in a particular geographical area, for example, within biodiversity hotspots, which enables the study analysis of the effects of environmental change on biodiversity and eventually producing a multidimensional and dynamic view of life on Earth. So these two strategies will be employed simultaneously by the project. Well, is it feasible? We have done and put the detailed calculations for behind this budget in the PNAS paper. I don't think things have changed significantly. If anything, the costs are going down due to advances in DNA sequencing. We have estimated that the total cost for delivering 1.5 million genomes over 10 years to be, in today's dollars, $4.7 billion US. Now, that might sound like a lot, but let's look at the cost of the Human Genome Project, sort of the first moonshot, if you will, in biology. The Human Genome Project in roughly $1993, $94, when the project was first funded, cost about $3 billion. And in today's dollars, that's the equivalent of $5 billion. So all of known eukaryotic biodiversity can be sequenced at less than the cost of sequencing the single human genome, 20 or so years ago. Now, does this sound like a lot of money? It still sounds, well, it's $4.7 billion. That's a lot of money. Well, $4.7 billion over 10 years is, on average, $470 million a year for the entire planet. That is, I think, reasonable. And when put in the context of other big science projects, for example, like the Large Hadron Collider. Okay, Large Hadron Collider in Switzerland cost $1 billion a year to operate, and it cost, the estimated cost for identification of the Higgs boson was $13.2 billion. So I'm not going to diss my physics friends and high-energy physicists, but I'll just ask, kind of what has the Higgs boson, done for you lately as compared to what 1.5 million genomes might do. And it's important to put this in perspective. This project has tremendous potential, not only to advance science, but to provide practical benefits for mankind. Okay, so I keep advancing somebody else's slides there, and I apologize for that. So we are attempting to sequence the eukaryotic part of the tree of life, which is everything but the purple and the brown there. Over all life, we estimate 2.3 million species. That is a tremendous underestimate because the number of microbes is vastly underestimated, maybe 3 trillion species. How do you even define species anymore in the archaeal and bacterial world? But this beautiful figure produced by Keith Crandall, and I'm sure we'll hear more about this in his talk, sort of summarizes what we know about sequencing of eukaryotic life at the genus level. As I mentioned, 1.5 million known, and today, in just another year and a half, when I first presented this information, we looked at this with John Coddington and Mike Trisma, we found 2,500 complete genomes in NCBI. Today, in just a year and a half, that has increased by 800. So we're now at 3,300 complete, quote unquote, complete genomes in the database. So there's 3,300, but let's unpack that a little bit. Let's look at that previous slide and with the phylogenetic wave, and let's look at the phylogenetic distribution of those 3,300. So yes, we have a representative of all the domains. We have a representative of the 5 kingdoms, but it was amazing to me that we only have representatives of about half of the phyla, the known phyla. When we started our California project, the California EBP, just by selecting 50 organisms, we selected 2 phylum firsts from a selection of 50 iconic organisms from the state of California. 50% of the phyla, less than that, 41% of the classes, a third approximately of the orders, and then of course the numbers drop off rapidly as we get to the numbers of sequence genera and species. So overall, if you look at the number of species, that's 0.2% of the known eukaryotic species. So everything that we know today in terms of genomic biology is derived from the knowledge of just 0.2% of existing biodiversity. So what do we have to learn from the other 99.8%? Well, let's first unpack this since we are here in London. And let's look at those 3,300 a little bit more carefully. Let's not just look at the phylogenetic distribution, but just look at the assembly qualities. And again, the resolution here may be a little bit different. This, let's just go on the x-axis there. If you look at 1, 2, 3, 4, the bottom row, that indicates how the genomes are described in NCBI. So the first number 1 is a contig assembly, the second is a scaffold assembly, the third is designated complete, chromosome, and the fourth is designated complete. And then after that is sort of a logarithmic system where the first number represents the log, okay, of the N50 value for contigs, and the second number there, the top number, represents the log N50 for scaffolds. So everything, well, maybe, everything here to the right, so these would be scaffold contig N50s of 1 million, and scaffold N50s from about here when you get to 10 million. About here is where the Vertebrate Genomes Project has placed their cutoff for reference genomes. So if you look at it, it's less than 100, really, of these 3,300 that would qualify as reference genomes today. Many of these genomes are quite good, and of course when you get to the smaller genomes, the fungi and so on, these are quite good, but these are, all these twos are scaffold assemblies. And so we would draw the line approximately here, and everything to the right here. So certainly less than half of the 3,300 we would say would be useful as a reference at this stage. So even though there's 3,300 listed, genomes, complete genome sequences for eukaryotes, probably only half of those are useful. So maybe 0.1%. So we are just really at the beginning, and we are learning. And there are several projects we're going to hear about today that are really teaching us the things that we need to know to scale up and produce reference genomes at high throughput, at scale, and at high quality. Okay, and the good news is, the costs keep coming down. And for reference genomes, the great news is the read lengths are going up. And the costs are coming down, the lengths are going up, and that means we're going to be able to really achieve high quality reference scale, high throughput reference scale genomes in a short period of time. An example of this was presented, and I'm sure Eric Jarvis is going to talk about this, a couple of weeks ago at the Vertebrate Genomes Project, where the first 14 of sequences were released, first 4 vertebrate sequences released, reference quality genomes, out of a total of approximately 260 ordinal level genomes that are planned for the Vertebrate Genomes Project. And these, I've been looking at these genomes, so everybody in the vertebrate genomes community knows I'm kind of a pain when it comes to looking at chromosome scale assemblies. And I am convinced that the genomes that we're getting out of the Vertebrate Genomes Project, which is easily transferable to all others, are the best genomes that I've ever seen out of the chute for any of the projects that I've looked at. These are really, really high quality assemblies made possible by a combination of technologies that I'm sure Eric's going to talk about. And of course, this has been complemented by the 25 genomes that were also recently released by the Sanger, Wellcome Sanger Institute just a couple of weeks ago for their 25th anniversary. So we're starting to see reference genomes produced at scale around the world, and we're going to hear about this for other taxa as well today. So today we're going to hear from those communities and projects across the world. It's going to be a very exciting part of our morning program, and we're really now in a great position with the Earth BioGenome Project to begin to coordinate our efforts so that we could reach our goals for phase 1 and the other phases of the project. And we do have a roadmap, and we're actually on time. We said that we were going to launch in 2018, and we are launching. We said that we would put into place agreements in the first year, and actually day 1 is tomorrow, but we already have 19 institutions that have committed to the MOU, as well as the 16 communities. Agreement on standards, again, this has been something that we have talked about from the beginning, but because of the fantastic work that's being done with the Vertebrate Genomes Project that Eric Jarvis leads at Rockefeller, I think we're already making great progress on developing the standards that are going to be necessary for EBP, and to stand up the organization. And those are the 3 things that we really need to do in the coming year, in addition to what we need to do to launch the actual sequencing effort, which is the collection, of course, the collection of specimens. And we'll be hearing from Katie. Is Katie here? And John about GGBN, and we'll be hearing about other collections of organisms for sequencing. So what are the goals of the workshop? Session 1, which follows my talk, will be to catalog progress of the affiliated institutional partners, large-scale and national and regional projects. Second, to assess the state of the art in sample acquisition, sequencing, assembly, and annotation of eukaryotic genomes. That will be the second part of the day in the afternoon. To present the scientific goals and desired outcomes in session 2. Also later in the day, the second half of this afternoon. To tomorrow, to evaluate relevant data use policies and to develop recommendations. We're going to have a panel discussion on that subject. Another panel discussion following to develop long-term strategies, a funding workshop for EBP and affiliated communities. And finally, in our last session, to really move forward to stand the organization up is to determine an appropriate governance model that we're going to use moving forward that goes beyond sort of the EBP working group that we have today. And finally, on tomorrow, we're going to announce, as most of you know, in the morning, the official day 1 of the project, the global launch of the EBP to the world, where we expect members of the international press as well as representatives from the major scientific journals to be here in attendance. So we are getting ready for liftoff for our moonshot for biology, and it is time that we go forth and sequence. So that's all I have for you in my opening comments.